Madayao. Good morning, everyone in the Philippines and around the world. Welcome to the second day of the Mindanao-wide Omics webinar with the theme, Omics in Mindanao, Challenges and Opportunities During the Pandemic. This two-day webinar series is presented to you by the Philippine Genome Center Mindanao in celebration of our third year anniversary. Today, we'll once again give you an exciting lineup of presentations from our speakers. We have Dr. 𝗢𝗺𝗀 𝗥𝗲𝘀, Vice Governor 𝗀𝗰𝘁 and Dr. Mark Lloyd Dapart. Yesterday, we showcased presentations from various experts in the field of omics, including the challenges and opportunities they faced or they experienced during the pandemic. We had Dr. Maria Elizabeth Naredo, Dr. Kay Ramos, and Dr. Joycelyn Humawan. Now, to give us a synthesis from the presentations of our speakers yesterday, please help me welcome the Program Director of Philippine Genome Center Mindanao, Dr. Joel Hassan Tolentino.
Hello, can you hear me?
Yes, sir.
Marianne?
Yes, sir.
Okay. Good day, everyone. Yesterday, we had a very interesting, informative, and insightful webinar. The first day of the 2022 Mindanao-wide Omics webinar, or Mindanao WOW, showcased the first half of the talks that convey the omics researches of our first three distinguished speakers and expound on this year's theme, Omics in Mindanao, Challenges and Opportunities During the Pandemic. But before we heard the presentations yesterday, the UP Mindanao Chancellor and former PGC Mindanao Program Director, Dr. 𝗀𝗪𝗪 welcomed the participants and opened the event, where she highlighted the critical role played during the pandemic by the then one-year-old PGC Mindanao, where the center provided capacity building and technical services in the fight against the epidemic. She stressed how the collaborative efforts and support and trust of the center's partners from the academe, government, private sector, civil groups, and media made the challenges faced by the center a reality. This was followed by special messages from UP System Vice President for Academic Affairs, Dr. Cynthia Bautista, and UP PGC Executive Director, Dr. Cynthia Saloma. Dr. Bautista reiterated the rationale of the creation of the Philippine Genome Center and its expansion to the Visayas and Mindanao. PGC was created as multi- and interdisciplinary institution that translates knowledge about genes and genomes into products that are made especially for Filipinos, for us to enjoy a better quality of life. She emphasized that PGC is first and foremost a Philippine center, meant to serve the nation, and UP only hosts PGC. Dr. Saloma, who attended live from Singapore, accentuated PGC Mindanao's involvement in the national COVID-19 biosurveillance program in partnership with SPMC, RESUS, and LGUs of Regions 11, 12, and BARM. Also, she provided insights on how COVID-19 molecular diagnostic laboratories that were established during the pandemic can still be useful after the pandemic is over. These facilities can be used for molecular diagnostic of other respiratory diseases and for precision health using nucleic acid-based testing, such as the early screening and identification of individuals who are at risk to develop diseases later in life. Now let's have the highlights from the talks of our three distinguished speakers. Our first speaker was Dr. Maria Elizabeth Naredo. from the Philippine Coconut Authority or PCA. Dr. Naredo's talk was entitled Genomics Research at PCA, Opportunities and Challenges Before and Beyond the Pandemic. She shared how they applied the DNA marker technology in the coconut hybridization program for the improvement of coconut variety, and how the use of gene expression analysis during coconut embryogenesis optimized its various stages in 𝗎𝑐. Dr. Naredo mentioned the advances in the sequencing technology in detecting variants of COVID-19 virus during the pandemic as an opportunity to be taken advantage by PCA in the 𝗪𝗪𝗪 viroid disease detection. Also, she showed the importance of sequencing of complete mitochondrial genome of coconut rhinoceros beetle, or CRB, in further differentiating the populations of this coconut pest. Finally, Dr. Naredo presented omics-related future projects of PCA, which includes genetic fidelity testing of plumule-derived coconut plantlets and phase 2 of 𝗎𝑐. Our second speaker was Dr. K. Ramos from the Western Mindanao State University. Dr. Ramos shared with us the conquered challenges and strengthened opportunities amidst the pandemic in genomics research in Zamboanga Peninsula. She shared the story of the birth of omics research in her university, which is through the establishment of a DOST NICER program on Zampen native chicken. Her project in this program aims to identify genes associated with economically important traits of Zampen native chicken through genome-wide association study. Dr. Ramos listed the challenges in the conduct of this research during the pandemic and how they conquered them. Among the challenges were in the procurement, delayed release of funds due to new protocols, the avian flu outbreak, and a fire incident in one of their molecular laboratories. Mitigating activities that enabled them to meet their deliverables despite the challenges include having partnerships or linkages with other institutions like the National University of Singapore and a very supportive administration. Conquering these challenges opened doors of opportunities. Our third speaker was Dr. Joycelyn Humawan from the Caraga State University. Her talk brought us to the interesting Lake Mainit, and it was entitled Transmission of Snail-Borne Trematodes in Lake Mainit, Philippines, probing the role of rice fields through a molecular perspective. Dr. Humawan's presentation enlightened us on the link of rice field exposure and neglected tropical disease such as schistosomiasis, the life cycle of the trematodes, including its definitive host and its intermediate host, the importance of elucidating at the molecular level the snail species found in surrounding rice fields of Lake Mainit, and the remaining gaps for omics research opportunities in this field. She shared the challenges their team encountered in the collection, documentation of the snail species, and DNA analysis during the height of the pandemic. These include travel restrictions, team members getting sick of COVID-19, and damaged samples for analysis due to prolonged closure of partner laboratories. Dr. Humawan emphasized the support of their technical partners like DOST and DOH Caraga, and acknowledged the roles of PGC Mindanao and Macrogen for the assistance in their molecular analysis and sequencing needs. These talks manifested that the pandemic has indeed challenged us in the implementation of our research projects. Nevertheless, many of these challenges made us more creative, collaborative, and resourceful in our project implementation and led us to discover and explore greater opportunities. Thank you, and I am sure the second half of the talks of our Mindanao WOW will still be very interesting, informative, and insightful. Good morning.
Thank you very much, Dr. Tolentino, for giving us a glimpse of our day one in the Mindanao-wide Omics webinar. We also thank our 101 participants in our Zoom. We also like to inform that we are also live via FB. So stay tuned because we will be giving out exciting prizes such as PGC Mindanao limited merchandises, and discounts on our training. So let's begin. To introduce our speakers and to facilitate our open forum, our moderator is a technical expert under the Research and Development Division of the PGC Mindanao. Let us all welcome Dr. Aleila Decadis.
Good morning, everyone. Thank you very much, Marian, for your introduction. So today we will be hearing another set of expert speakers, who will be delivering their lectures on their work on omics. So our first speaker, video please, is Dr. 𝗀𝗀𝗀𝗪 She is a molecular biologist, a virologist who has worked with viruses such as HIV-1, bovine leukemia virus, and SARS-CoV-2 over the last decade. She received her bachelor's degree in biology from the University of the Philippines, Mindanao, and obtained her master's degree in biotechnology from the University of Tokyo, Japan, under the Panasonic Scholarship. She then pursued her doctorate in biotechnology with a specialization in virology from the same university under the Honjo International Scholarship Foundation. Thereafter, she served as a postdoctoral researcher at the Institute of Physical and Chemical Research, or RIKEN, also in Japan for almost 2 years, and currently she works at Trinity College of Dublin, Ireland as a research fellow. Dr. Onggo's work as a molecular virologist over the last decade was focused on molecular biology and virology research projects. As a PhD student at the University of Tokyo, she performed functional studies on lentiviral proteins, developed a high-throughput screening assay for HIV-1, and used this assay to screen a small molecule library. She discovered potent drugs that inhibit a viral host interaction in HIV-1 replication. During her stay at RIKEN, Japan as a postdoctoral researcher, she worked extensively on understanding the mechanism of actions of these drugs in inhibiting HIV-1 replication. She also worked on the generation and characterization of cell lines stably transfected with bovine leukemia virus that is essential for BLV vaccine development. Her current research work at the Trinity Biomedical Sciences Institute of Trinity College Dublin is focused on investigating the antiviral immune evasion mechanisms of SARS-CoV-2 in epithelial and immune cells. So to talk about elucidating antiviral immune evasion mechanisms of SARS-CoV-2 using omics approaches, lessons for the future, let us welcome Dr. 𝗀𝗀𝗀𝗪𝗀𝗪𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀 we have a record. He has a recorded video. Jan, thank you. Morning, Weng. It's 2 a.m. in Dublin. Thank you very much for waking up this early.
Good morning. Yes.
In
It-
celebration of PGC Mindanao's founding anniversary, I'd like to commend you for the great things that you've accomplished in the past 3 years. I've been involved in the study of SARS-CoV-2 since last year. I was given the opportunity to work at Trinity College Dublin to investigate the antiviral immune evasion mechanisms of SARS-CoV-2. I will discuss a little bit about my study later in the next few slides, but I will also discuss the mechanisms known so far, mechanisms of SARS-CoV-2 in immune evasion, as well as the different approaches used to identify these mechanisms, including omics approaches. It is important to understand how viruses evade the immune response because this will allow us to identify antiviral drug targets. This will identify specific step in the innate signaling that are targeted by the host, the viral proteins. With that information, we'll be able to devise novel antiviral treatments or strategies, or even repurpose our existing drugs to also allow us to develop vaccines or redevelop our existing vaccines with probable deletions at the viral genes required for host immune suppression. And eventually, this will help us or allow us to curtail the transmission of the virus. Our innate immune response is our first line of antiviral response and efficiently inhibits viral replication through the production of interferons. These are antiviral factors secreted from infected cells, which activate immune cells to produce cytokines and express antiviral genes such as interferon-stimulated genes. Firstly, the SARS-CoV-2 or the molecular patterns from SARS-CoV-2 is recognized by proteins collectively called as pathogen recognition receptors or PRRs. Activation of PRRs will induce the signaling cascade or the activation, the signaling cascade that will produce interferons. Production of the produced interferons will bind to its respective receptors that are found in the plasma membrane of uninfected immune cells. Binding will trigger the activation of the host cellular pathway called the JAK-STAT pathway and phosphorylate the proteins, and that will lead to the expression of antiviral genes called interferon-stimulated genes and pro-inflammatory genes that are used to target and eliminate the virus. However, we know that SARS-CoV-2, as of now, we know that SARS-CoV-2 is a master of immunization. It has developed or evolved several mechanisms to counter the innate immune response. These the mechanisms used by the virus to counter the innate immune response have been identified as of this time, and they are categorized into 4 mechanisms. The first one is the evasion of sensing by the host innate immune receptors. So the SARS-CoV-2 developed strategies to avoid sensing of our innate immune receptors through formation of DMVs or double membrane vesicles, through RNA capping, or through inhibition of the receptor PRR RIG-I by N protein. The second mechanism is inhibition of innate immune receptor signaling and interferon production. So the step following the recognition that will lead to the production of interferon. So the viral proteins would prevent, for example, the phosphorylation of IRF3, and thereby it prevents the nuclear translocation of IRF3. It would lead to the production of interferon. The third step is the inhibition of interferon signaling and the expression of ISG. So this is inhibited by the virus by preventing or blocking the binding of the interferons to its receptors, or through preventing the phosphorylation of the STAT proteins, thereby inhibiting ISG expression. And the fourth mechanism is the inhibition of host protein production. So for example, NSP1 prevents the nuclear export of mRNA, host mRNA, while NSP8 and NSP9 prevent protein trafficking. These mechanisms were identified using several studies, and within those studies there are several methods. So these studies include overexpression studies, protein-protein interaction studies, RNA-RNA interaction, RNA-protein interaction studies. These 3 constitute the virus-host interaction studies, as well as reverse genetics and other methods that I may not be able to discuss here. So, but prior to this, the identification of the first SARS-CoV-2 genome laid out the foundation of the study of the virus, as well as the different aspects of including the immune evasion mechanism of the virus. This was identified through metagenomic next generation sequencing, wherein all nucleic acids in a sample are simultaneously and independently sequenced and assembled as contigs and are aligned to a reference database for classification. Through this and subsequent studies, we now know that SARS-CoV-2 encode for or contains polyprotein ORF1ab, which encodes for non-structural proteins NSP1 to 16. And it has structural proteins, spike, envelope, membrane, and nucleoprotein, and in between those are 9 accessory proteins. A study using viric RNA sequencing found subgenomic RNAs in the 5 accessory proteins, the 3A, 6, 7A, 7B, and 8. These RNAs are essential for the translation of the structural proteins and the accessory proteins. The method employed was direct RNA or nanopore-based RNA sequencing from samples from RNAs extracted from infected monkey cells. Then for the studies identifying the mechanism of viral immune evasion, we have gene overexpression study. This is actually a commonly performed studies to characterize the function of a known gene. For example, here the viral gene of interest is cloned into an expression plasmid and introduced into a eukaryotic cell, a mammalian cell, to identify its phenotype and localization and eventually its functional role. One study identified the impact of the SARS-CoV-2 proteins in the 3 branches of innate immunity, the virus sensing, cytokine signaling, and autophagy. So they expressed or introduced the SARS-CoV-2 proteins in a mammalian cell and used reporter assays such as luciferase assay to determine its effect in each of these pathways. They were able to identify which specific proteins target the autophagy, virus sensing, and cytokine signaling. For the virus-host interaction studies, there are several methods that could help us investigate the interacting partners. If it's like RNA, we have several advanced methods. Also for protein-protein interaction, we have tandem affinity purification and co-immunoprecipitation, which is coupled with the mass spectrometry. And protein RNA interaction, usually coupled with RNA sequencing and mass spectrometry. So RNA interaction studies, for example, in this study, they were able to identify 300 host RNAs that interact with the SARS-CoV-2 genomes, while protein interaction studies identify the specific proteins or host proteins that interact with each of the SARS-CoV-2 protein. And we have RNA protein interaction, which identify the host proteins that interact specifically to the viral RNA. To highlight one of the methods from protein-protein interaction, we have tandem affinity purification mass spectrometry, or TAP-MS, which was used to generate this protein-protein interaction network. So the SARS-CoV-2 proteins were cloned and then strep tagged and expressed in mammalian cell line, and they perform affinity purification and mass spectrometry to identify the bound host proteins in each of the SARS-CoV-2 protein. And for RNA protein interaction, this result was generated from three methods employed from these studies, which has four common steps, which includes, firstly, the isolation of the RNA protein complexes from the infected live cells, followed by the purification of the complex through cross-linking and the elution or the capture of the complex, and RNA digestion, followed by proteomic analysis to identify the proteins that bind to the viral RNA. And we have another method, which is a powerful one, that would allow the study of the viral proteins in the whole virus context, because you have these, the reverse genetics here utilizes an infectious clone generated from the full length cDNA of the virus, and you can induce a mutation in this clone, and then conduct a study to identify its phenotype in terms of virulence or transmission, or its ability to inhibit interferon production or induction, interferon induction and action. So with that, we already know a lot of information about the mechanisms of SARS-CoV-2 for immune evasion. But we can still dig deep, we can still study further. In our case, we are interested in studying inhibition of interferon signaling and ISG expression by viral hijacking of the E3 ligase host proteins. Previous studies in the lab identified a binding motif called suppressor of cytokine signaling box, or SOX-box binding motif from respiratory syncytial virus, hepatitis C virus, and HIV, which which binds to the proteins elongin B or elongin C, RBX or COL, that are involved in the formation of the E3 ligase complex. So upon binding, this would promote ubiquitination of the SAT proteins and thereby promote its degradation. And thus, the interferon signaling is inhibited, and thereby inhibits the expression of the ISGs or antiviral genes. These through bioinformatic analysis, this binding motif was also identified in SARS-CoV-2 NSP14, and we hypothesized that the mechanism or the SARS-CoV-2 induces host protein degradation by proteasomal activity. To confirm this, we are conducting transfection and infection studies using wild type version of the virus or the gene, as well as the modified version with the deletion or mutation in the NSP14 protein. And we induce the interferon signaling with interferon alpha at different time points, and extract the genetic material and conduct several analyses, RNA sequencing for the expression of ISGs, and qRT-PCR for the expression of the the components of the JAK-STAT pathway, as well as Western blotting analysis for the proteins involved in the JAK-STAT pathway, and ELISA for the production of cytokines. Initial challenge was to identify or to find or to create an infection model. So it was a bit difficult to identify first which epithelial cell line is suitable or susceptible for SARS-CoV-2 infection. So we are lucky that we obtained epithelial cell line that has ACE2 and TMPRSS2 entry receptors for SARS-CoV-2. So we found it to be more susceptible for viral infection as viral production is higher than this other epithelial cell line. And we have also confirmed its protein and protein expression and the viral gene expression. Personally, for me, it's one of the challenges is to breathe through this respirator while working for about 2 to 3 hours in the BSL-3 laboratory. But as a group, one of the challenges is to get the license from Environmental Protection Agency for the use of the genetically modified strains. We have to address the questions from the public as we inform the public through advertising it in local newspaper of our intent to use the GMOs, and we have to address the questions from the Environmental Protection Agency. We already obtained the license, but we have to adhere to the license conditions, change of laboratory setup and workflow, and we are restricted, or the immunocompromised staff as well as pregnant women are not allowed to work in BSL-3. And another angle to be looking at is the effect of the SARS-CoV-2 in interferon pathway using the immune cells. So we have COVID-19 infected blood samples from COVID-19 infected patients and healthy controls, and we isolated their immune cells, the PBMCs and neutrophils, and induced interferon signaling through interferon alpha at different time points, and similarly obtain the genetic materials for different analysis that are of the gene expression and protein expression and cytokine production that are involved in the interferon signaling. And we are also looking at the gene overexpression from the using the SARS-CoV-2 proteins. We transfected the each individual construct that encodes the viral protein to epithelial cell line and conduct gene expression analysis and protein analysis. We have confirmed its overexpression in epithelial cell line as well as its protein expression. And interestingly, we found that there are viral proteins that upregulate our host protein of interest, SOX proteins. The SOX proteins were found to upregulate also or upregulated in other viruses such as influenza virus, HSV1, EMCV, and VV. When it was upregulated, it was found to enhance the infection and inhibit the interferon signaling. So we'd like to explore further whether the proteins that upregulate the SOX proteins, when inhibited, or when using knockout cell lines of the SOX proteins, we want to know if these restore the interferon signaling. There are a lot to conduct. There are a lot of experiments to conduct, and we are still at the preliminary stage, so I can't show you our data yet. So it's because it's preliminary, but yeah, hopefully we can accomplish this one in less than 2 years. So in summary, we know that it is important to understand how SARS-CoV-2 escape immune response so that we can design strategies to combat COVID-19 disease. And we know now that SARS-CoV-2 uses different mechanisms to inhibit every step of the immune response using it individual or using its viral proteins. And we have several methods, including omics approaches, which we can use to study virus-host interactions, which will help us unravel the viral immune evasion mechanisms. So I'd like to thank members of the laboratory, especially my principal investigator, for giving me the opportunity to conduct this study, as well as the collaborators for providing us the strains that we need for the constructs that we need for our study, and our funding agency, the Science Foundation Ireland. Thank you so much for listening.
Thank you very much, Wang. We are very proud of you, coming from UP Mindanao. So ngayon you're doing your extensive research on SARS. So thank you very much for your detailed discussion on how SARS-CoV-2 escape our immune system during infection. So while our audience is preparing their questions, I know this is very technical, very basic research, but I hope you can come up with a very good question for our speaker, our fellow Mindanaoan. So let me introduce our next speaker. So please stay put, Ma'am Wang. I know you are awake, It's 2 a.m. in Dublin. So our next speaker is Vice Governor 𝗪𝗀𝗀𝗪𝗀. He is the current Vice Governor of the province of Davao de Oro. He officially assumed the Vice Governorship last July 1, 2022. Prior to that, he served as the Governor of Davao de Oro from 2016 to 2022. In his stint as governor of Davao de Oro, it was hailed as one of the most business competitive provinces in the Philippines, an emerging tourist destination in Mindanao, and was proclaimed an insurgency-free province. As governor, he also served as the chair of the Regional Development Council of Davao Region. He led and steered the RDC-11 in addressing the pandemic and its impacts through the creation of the Regional Task Force Against COVID-19 in March 2020. The crafting of the Davao Regional Recovery and Resiliency Program to mitigate the impacts of the pandemic to the economy, and the expansion of SARS-CoV-2 biosurveillance project in Mindanao. In 2017, the Civil Service Commission hailed him as one of the most outstanding government workers in the entire country. The Benita and Catalino Yap Foundation awarded him also as one of the innovators of Asia. In 2021, the Department of Science and Technology awarded Vice Governor Tyrone with the Science for the People Award for his groundbreaking scientific projects and programs that improve the quality of life of the people of Davao de Oro. Vice Governor Tyrone Uy finished a degree of medical technology from Veles College in Cebu and a master's degree on public administration from the Ateneo de Davao University. So to talk about innovation, creativity, and change amidst COVID-19, notes and insights from Davao de Oro, let us welcome Vice Governor Tyrone J.V. Uy. Good morning po.
Good morning po, ma'am. Good morning po sa ating lahat. Can you hear me?
Yes po.
Klaro
Yes
ba
po.
yung reception
Yes
ng
po.
linya natin? Okay.
Yes po.
So, thank you so much for the long introduction, ma'am. Baka ma-short pa yung salita ko. Anyway, thank you so much for inviting me for today's event. It is my honor and pleasure to speak in front of you all. I'm gonna talk about, my topic is about innovation, creativity, and change amidst COVID-19. Davao de Oro experience. No? So magandang umaga po sa ating lahat. Good morning as we say it in Davao de Oro. It is an honor to speak before sa inyong lahat. I admit that I was surprised upon receiving the invitation from the Philippine Genome Center of Mindanao. Nagpapasalamat kay ating Mindanao Chancellor ngayon na inimbitahan po ninyo ako. I thank also the PGC Mindanao for the chance to relate my story today sa ating mga stakeholders. And I will try my very best to share you our best practices as so far as being the former governor of Davao de Oro sa provincial local government unit namin, no, dito sa probinsya namin. Sort of a background, Davao de Oro is the northernmost part of Davao region. Our province is a gateway to Caraga region or the south of Mindanao, specifically Davao Oriental. No? It's formerly known as 𝗠𝗶𝗀 Valley, but was officially renamed to Davao de Oro last 2019, shortly before the pandemic started. No? December of 2019, yung plebiscite natapos, and we garnered 97% of our majority to vote yes the renaming of our province to Davao de Oro. It was 23 years of existence. Davao de Oro is no stranger to crisis. If everyone can still remember, in 2012, our province suffered the worst of the Typhoon Pablo. That is the tragedy that no one thought would happen. No? Kahit sino sa mga taga Davao didn't expect that to happen. No? Na kumbaga lumaki tayo na ang ating idea of Davao region is being shielded from the major typhoons because we have marami pa tayong mga bukid to protect us. But then it happened. No? The local government, through the help of the private sector and the national government, shepherded the province through those difficult days. From the preparation phase, retrieval operations, up to the recovery and rehabilitation phase stages. Thank God naman na nakabangon na tayo noong panahon ng Typhoon Pablo. My baptism of fire came last 2020. In a snap, the entire country was placed under state of public health emergency. And I would speak in behalf of the local government units when I say that we are caught unprepared. Totoo naman, hindi naman tayo talaga preparado sa ganon ka-wide scale na problema, no, ng pandemic. And it was a worldwide phenomenon that we experienced together until now. So our country is used to typhoons, earthquakes, even armed conflicts. Alam natin yan. But a pandemic is something we have not anticipated for. Yun ang problema. So we do not have models or templates on how to approach the pandemic. The recent public health crisis with the same magnitude as the COVID-19 happened a century ago pa. As a result, it brought our already weak healthcare system to its knees. Hirapan talaga tayo. And as a consequence, ang economy natin nasakripisyo din dahil sa sunod-sunod na mga lockdowns during that time. Our movement across the regions, across the provinces for trading has also been halted. So, moreover, the public health crisis triggered a lot of damage in the economy. It accelerated and compounded our vulnerabilities. Because healthcare is a devolved function of the LGUs, it means that LGUs are also the frontliners of responding and managing the public health crisis. This response and management, however, is seriously crippled by the limitations of the LGUs in terms of financial and human resources. But hindi ko in-accept yun na excuse. No? Ang ginawa namin or ang nasa isip ko, crisis can also be an opportunity for innovation. Diba? That's human nature to us. When pushed to the wall, mas marami tayong na nasa isip na ginagawa natin. So, kumbaga, kapag survival na ang iniisip ng isang tao, you really have to do something. No, that's our human nature. Tapos, opportunity for creativity and opportunity for change. So, importante na ma-imbibe natin sa ating mga constituents na pwede palang kumbaga gamitin natin itong sakuna or crisis to our advantage. So putting the people first. The first thing we did is to make clear that our priority is to put the people first. So importante, safe yung tao natin. Upon the announcement of the first COVID-19 case in Davao Region, swerte pa nga kami dahil sa amin talaga yung first case sa Davao de Oro, taga-Pantukan na tayo, taga-Pantukan. We activated our Emergency Operations Center and converted our 4 provincial hospitals into COVID-19 emergency hospitals. We mobilized our health emergency response teams in all municipalities and barangay. We also held series of consultative meetings with all the sectors involved. We acted with urgency, employed scientific and evidence-based policies, made sure that everyone gets on board through the whole of society approach. And we followed a simple yet sensible strategy. The H2H or household to hospital strategy. Through the Sangguniang Panlalawigan, we mandated the observance of the minimum health standards. Until now, we also prepared our community isolation centers in the barangays and the municipalities to contain the virus. In a matter of weeks, Davao de Oro has 285 barangay isolation units and 17 municipal isolation centers over time. As soon as we were able to establish isolation units where our patients are monitored and treated, a problem in testing emerged. Diba? Ito talaga yung point where dapat ang gobyerno palakasin talaga ang testing dahil wala tayong makukuha Hindi natin alam sino yung kalaban natin. Hindi natin makita. But through testing, we were able to identify and isolate our yung mga natamaan. In terms of swab test, Davao de Oro is heavily reliant sa DOH, no, or sa mga hospitals like DRMC of Tagum City and the Southern Philippines Medical Center or the SPMC in Davao City. Kaya lang, nang na-experience natin, matagal masyado yung downtime sa result. No? Maghintay ka ng more than a week para lang sa result. So, hindi talaga siya acceptable during that time. No? Hindi accurate yung data natin para ganon. So, hindi rin accurate yung pag-intervene natin sa mga pasyente. No? So, yun ang problema. The gap in testing resulted to a delay release or transmission of results to local governments, which in turn resulted to prolonged isolation of suspect and probable cases. Overburdened testing labs. Nagpilahay na ang mga LGUs kay daghan ng uli. Gikan sa Manila, gikan sa Cebu, gikan sa Bacolod, gikan sa laing mga lugar. No? And then the hospital staff. Davao de Oro is only given 100 slots per day to test the suspect and probable cases. Tapos that time, ang policy sa gobyerno, basta expose ka, you have to contact trace. So maraming mga sanga-sanga yan and they have to be tested. So yun ang problema. This is where the idea of constructing our own COVID-19 testing biomolecular laboratory started. I learned about the Philippine Genome Center and University of the Philippines, Mindanao, through Facebook post of Assistant Regional Director and DOST 11, si Sir Kenneth Baroga, and I messaged him kagad, no? And a series of meetings and discussions were set between the PLGU of Davao de Oro, DOST, Philippine Genome Center. Binayin na rin namin ang Department of Health and of course the UP Mindanao in general. When we were contemplating of constructing our own COVID-19 testing laboratory, the national government through the Bayanihan Act gave all the provincial government funds to do it. Without hesitation, we decided to allot the money in the construction of the testing laboratory. On June 22 of 2020, a memorandum of agreement between us and the Philippine Genome Center were entered into. Together with me are the chiefs of our hospitals, of the 4 provincial hospitals, then PGC Mindanao Director Dr. 𝗪𝗁𝗁𝗍, and then UP Mindanao Chancellor 𝗪𝗁𝗁𝗍, attended and signed the memorandum of agreement doon sa partnership natin with them. We sent our in-house medical technologies and other hospital staff to UP Mindanao for trainings in the PGC. And on February 26 of 2021, the Department of Health formally issued our license to operate. And by March 5 of 2021, our own biomolecular laboratory was inaugurated in Davao de Oro Provincial Hospital in the municipality of Monte Vista, Davao de Oro. So together with us are the officials from the Philippine Genome Center of Mindanao, Department of Health, DOST, and others. And our testing laboratory is the first and only COVID-19 testing laboratory run and operated by a provincial hospital in Davao Region. Because of the biomolecular laboratory, we were able to address several issues and problems that emanated from the gap in testing. Prior to the establishment of our COVID-19 testing laboratory, probable and suspect cases will have to wait for 5 to 7 days in their respective isolation centers before getting their official and confirmed results. Thus, prolonged isolation become the reason why the municipal isolation centers are overcrowded and reports of people facing mental health issues while in isolations are getting rampant. Lalo na rin nagakamot ang ulo ng mga LCE dahil the more andyan yung mga patients sa ating mga isolation center, syempre gumagastos din ang ibang mga local government sa kain nila and their other needs, no? So dagdag expenses yan siya. So this is where we have to, kumbaga, yun ang game changing na nangyari sa amin sa Davao de Oro. Because of the molecular laboratory, we were able to address several issues, no? And this prolonged isolation, yung sabi ko kanina, many people or some people also complain that when they are kept in isolation for longer days, they also lose their income, no? As a result, isolation also has its economic impacts. When the COVID-19 testing laboratory operated, it was reduced to 1 to 2 days. So dako kay diferensya. From 100 specimens that are tested every day, we are now able to test as much as 500 specimens every day. To better operationalize our testing system, assisted by our own COVID-19 laboratory, we also made our very own COVID-19 testing information management system. We're able to facilitate, record the management of COVID-19 conducted tests in the testing lab. We also generated the COVID-19 test result report with security features and provide validation on the release COVID-19 test results. Our coordinations with our RHU units was fast-tracked through the COVID-19 laboratory. And by mid of 2021, series of surges happened. This is when the most contagious variants such as Omicron and Delta hit the country. The laboratory helped us in coping with the increased number of people who needs to be tested. We are not reliant anymore to the other institution in terms of testing, and we are able to help the testing capacity of Davao region. DRMC and SPMC can now extend more focus to localities that needed their help the most. And pasalamat din kami sa DOH, no? Kasi nag-counterpart din sila sa pag-produce sa mga reagents and the testing kits. Nagbigay din sila sa atin galing sa National Task Force COVID. Looking back, there are people who questioned my initial decision to set aside money for the construction of COVID-19 laboratory. Because noong 2020, uso talaga yung bigas ang pinaminigay. No? Mga dole-outs. No? Uso yan. But for me, ang pinili ko is the COVID-19 laboratory, which is, for me, mas malaki ang impact and mas malaking maitutulong in the coming years, especially, alam natin, hindi pa huli lahat. Hindi pa ito ang the last pandemic that we'll be having. In the future, siguro, meron pang mas malala, no? Or marami pang mga ganitong situation ang ma-experience natin. So, at least ready na tayo. There are those who suggested that I should have channeled it more, no, sa mga isolation and treatment centers also. I acknowledge their points of view and their perspectives are certainly very valid. But I was looking for a long term. Without testing, how can we distinguish the infected from those who are not? How can contact tracing and treatment happen if we cannot separate the infected from the non-infected. I know the value of increased testing capacity on a personal level. I finished medical technology in undergrad, and I have also worked on clinics and laboratories. As I always say, we cannot see the virus unless we test them. Now, the COVID-19 laboratory in Davao de Oro can further aid the province in terms of research and development, especially in partnership with the Philippine Genome Center. The COVID-19 will not be the last of the pandemic of our lifetime. The biomolecular laboratory will serve as the Davao de Oro's first line of defense and layer of protection if that time comes. Before I end my presentation, allow me to share with you our other initiatives in mitigating the COVID-19 pandemic. To make the healthcare services in the outpatient department unhampered in our 4 provincial hospitals, we created the Call the Doc program, your virtual doctor program. Patients in the OPD need not to personally show up in the OPD clinics. They can already receive free teleconsultation through text, chat, or calls. This allowed our hospitals to continue to serve the public, at the same time enforce social distancing restrictions more effectively. Because the healthcare services are now available online, less patients are already coming to the hospital, thus decreasing the chances of the COVID-19 transmission too. No, naging advantage ito sa ating mga doctors because I put premium to the health of our healthcare givers. Dahil konti na lang nga yung doctors natin, konti na lang nga yung mga nurses natin and medical staff. Kung matamaan pa ang hospital, ang mga workers natin ng COVID-19, patay. Sino na ang mag-take care sa mga pasyente natin na nagkakaroon ng COVID-19? So, this is a program that addresses that concern. The program also was hailed by the Department of Information and Communication and Technology and the Department of Interior and Local Government as one of the most innovative COVID-19 response programs in the entire country. The Galing Po Awards also shortlisted this program to be one of the best LGU programs during the pandemic. Another innovations we introduced is the DDO helplines. Through the helplines, our constituents need not to go to the local government offices and national government agencies to avail of the government services. Pwede na sila mag-communicate through chat, calls, or text messaging. The DDO helplines became a digital government center where clients can contact relevant government agencies to ask help during the pandemic. Digital innovations, namely the blood distribution management system, the DDO portal, customer service feedback and complaint, DDO QR system, and online recruitment. And also the vaccination management system are also introduced. We also introduced food security measures during the pandemic, such as Farm to Frontlines initiative. The PLGU brought all the produce of the local vegetable farmers nung panahon sa mga lockdowns na hindi sila makaka-deliver sa kanilang mga market, kaysa malata yung mga gulay, kasi we have one of the best vegetable farms in Davao region, binili namin at dinistribute na lang namin sa mga frontliners namin na almost 20,000 during the time, from our medical staff in the hospitals up to the barangay functionaries or yung create natin na mga barangay health and emergency response teams members na binigyan natin ng mga gulay at prutas para magamit nila sa kanilang pag-serbisyo sa atin, no, during that time. We also introduced the online bagsakan and the DDO shop local to connect our local industries to potential markets in the province, no? Sa mga bata din, to help our schools during the implementation of the distance learning modalities, we implemented the Radio Z Eskwela, TV Eskwela, and implemented our school-based feeding program utilizing the central kitchen approach to continue the provision of free lunch meals to school children. No, distribute pa namin ito sa mga purok-purok, no, during that time na wala pa tayong face-to-face learning. So we did not deprive our children to the right to food, no, during this time in the pandemic. So we were also guided by the Department of Education on this. So all of this we're able to achieve through partnerships and collaboration. My conclusion is that what we lack in terms of resources, we compensate through our active networking to potential agencies and organization na pwede makatulong sa atin. Looking back, I did not regret the one message that I sent to Sir Kenneth Baroga of the DOST. Kasi ito ang nag-lead sa ating partnership with the UP Mindanao and the Philippine Genome Center, which in turn help us in the building in our own COVID-19 testing laboratory. It is also important for local governments to work with science and technology sector, ang academe, because they also possess knowledge and resources that we can maximize all together. The innovations that we introduce to mitigate the impacts of the pandemic, from the increase in our testing capacity to ensuring food security and continuity of public service, attest to the enduring power of cooperation amidst crisis. It is when we work together that we get to be creative, innovative, and we allow positive change even in the world's difficult days to happen. So I hope na naamoy natunan sa Davao de Oro experience sa panahon na ako pa ang gobernador. And I would like to thank again the institution of the University of the Philippines through Chancellor Mamurao sa imbitasyon na gihatag nimo sa APUA o sa paghatag o higayon na makapaambit ta sa ato ang mga natun-an o sa ato ang mga best practices gikan sa among minahal na bulawanong probinsya sa Davao de Oro. Salamat ug maayong adlaw kaninyong tanan.
Thank you very much po, Governor for collaborating with PGC Mindanao and of course with UP Mindanao in handling this COVID-19 crisis. Indeed, the partnership between the academe and the government is needed, especially during this pandemic. Please stand by po for the open forum after our last speaker. Thank you po, Vice Governor.
Thank you, ma'am.
Thank you po, sir. Let me turn over the spotlight to Marian for our games. Surprise ito, mag-join mo, ha?
Thank you, Doc Alila, for introducing our first two speakers for this morning. We also thank our 141 Zoom participants for this webinar, as well as our participants who are joining us via FB Live. So it's time for our question and answer game. The first person who can type the correct answer in the chat box will be the winner. Are you ready? Here we go. Give one importance of understanding virus immune evasion. the answer is any of the following: to identify antiviral drug target, to devise novel treatments or strategies, to develop vaccines, and to curtail the transmission of the virus. So, who's the winner? So the winner is Mr. 𝗪𝗀𝗀. Congratulations, sir. You won 5 customized limited edition PGC Mindanao merchandise. One of our staff will get in touch with you for your prizes. So stay tuned later because we will have another game. Mama Leila, will transfer the screen to you.
Thank you, Marian. So, sinong nanalo? Sige. Okay, let's proceed to our last speaker for today. So, our last speaker is Dr. Mark Lloyd Dapar. He took his BS General Biology in Mindanao State University, Iligan Institute of Technology as magna cum laude. He also finished his biology in the same university and awarded the Gawad Chancellor sa natatanging mag-aaral sa larangan ng pananaliksik at haynayan during his BS Biology in 2015 and MS Biology in 2017. In 2020, he finished his PhD in Biology, major in Plant Sciences at the Pontifical and Royal University of Santo Tomas, Manila, also as a magna cum laude. He was also privileged to be invited as a junior researcher for a 6-month research fellowship in ethnopharmacology and molecular systematics at the Universität Bayreuth, I hope I pronounced it correctly, Dr. Mark, in Bayreuth, Germany, sponsored by the Alexander von Humboldt Foundation, a prestigious sponsorship for postdoctoral and experienced researchers of all nationalities in Germany. He was deeply honored to contribute 20 book chapter publications featuring 20 endemic, iconic, and interesting medicinal plants in the Philippines to the largest book project on the ethnobotany of the mountain regions of Southeast Asia by Springer Nature Switzerland in 2020. Currently, he is an assistant professor in the Department of Biology, College of Arts and Sciences in Central Mindanao University. He is also the University Coordinator in Microtechnique and Systematics Laboratory of the Natural Science Research Center and the University Coordinator in Biodiversity Education Training Unit of the Center for Biodiversity Research and Extension in Mindanao. Dr. Dapar is a young, vibrant, and prolific researcher doing participatory research engagement to study and save the rich ethnobotanical, ethnomedicinal, and ethnopharmacological knowledge of more than 100 ethnolinguistics groups in the Philippines. So to talk about integrative molecular taxonomy of medicinal plants used by indigenous peoples and cultural communities in Mindanao, Philippines, let us welcome Dr. Mark Lloyd G. Dapar. Good morning, Dr.
Good morning.
Grants, can you play Dr. Dapar's presentation, please?
Good morning. Isang mapagpalang umaga po sa ating lahat. Mayong buntag and happy 3rd year anniversary PGC Mindanao. And welcome to my webinar talk on the integrative molecular taxonomy of medicinal plants used by indigenous peoples and cultural communities in Mindanao, Philippines. To begin, let's take a look at the global perspective. The World Health Organization, which is very busy right now as the specialized agency of the United Nations responsible for international public health, is continually promoting TCAM, which means Traditional Complementary and Alternative Medicine. And as a matter of fact, 60% of the world's population rely on traditional herbal medicine, and of which 80% of the developing countries depend almost completely on traditional medical practices, particularly herbal remedies for primary health care. And we are so blessed in this planet Earth with 35,000 to 70,000 medicinal plant species, with 7,000 medicinal plant species in South Asia, comprising 6,500 medicinal plant species in Southeast Asia, and of course our very own country, the Philippines, with 1,500 medicinal plant species since 2004 and still counting. Now let's magnify our very own country, the Philippines. The Philippines is not just mega diverse in biodiversity but also ethnicity, among indigenous peoples embracing more than 100 divergent ethno-linguistic groups. It is renowned as one of the mega-diverse countries around the globe and one of the world's 8 biodiversity hotspots. It is also acclaimed to be diverse in tribal communities classified based on its unique identity, language, socio-political systems, and practices. And there are 120 medicinal plants in the Philippines scientifically validated for safety and efficacy. And the top 100 best medicinal plants in the Philippines were already listed. And so the rich knowledge, ecological understanding, resource management, and conservation practices of the locals and the IPs are actually recognized as imperative partners in environmental management because they act as stewards of their ancestral territories. And because of that, based on the United Nations Sustainable Development Goals, while we are targeting to reduce poverty, promote good health and well-being, it is imperative to reduce inequalities of IPs and involve them in drug discovery and development to achieve stronger partnerships for SDGs. And because of that, scientists nowadays are tapping the unique expertise of the IPs. And among the settlements of these IPs, the island of Mindanao is largely occupied with various IPs, with 61%, followed by Luzon with 33%, and the rest Visayas with 6%. So here, I have enlisted a total of 25 published ethnobotanical studies on tribal communities to date. And here, let's take a look in Mindanao with the largest number of IPs. First, we have the Muslim Maranao. While Maranaos are considered not among the Lumads or indigenous peoples, they are classified as a Moro group or a religious group with cultural practices. So they are considered to be cultural communities. We also have the Higaonon community in Rogongon, Ligan City, the Subanon communities in Ezami City, in Dumingag, and Lapuina in Zamboanga del Sur. We also have the Talaandig community in Valencia City, Bukidnon, Matigsalog community in Marilog, Davao City, the Balan community in Mount Matutum, South Cotabato, the Tagabawa community in Santa Cruz, Davao del Sur, the Tiguahanon Manubo community in San Fernando, Bukidnon, the Higaonon community in Agusan del Sur, and my recent expedition among the Agusan Manobo community in the province of Agusan del Sur. And here we are proud to present our review article that gives an account of the current status, challenges, recommendations, and future perspectives of Philippine ethnobotanical studies on indigenous communities. And you can also check here what are the other indigenous communities left undocumented. And now let me share you briefly about the Agusan Manobo. Etymologically, Manobo originated from the word Mansuba, which is a combination of man meaning people and Suba meaning river. They are located in the mountain ranges and hinterlands of Agusan del Sur, near Agusan River Valley and Agusan Marsh Wildlife Sanctuary. Their livelihood varies from rural agriculture or slash-and-burn method, fishing, hunting, and animal husbandry. Their dialect is known as Minanubo, and their traditional medical healers are known as the Babaylan, who can talk to a deity or spirit known to be 𝗪𝗓𝗄𝗧. And their common community plant resources varies from medicinal, ornamental, economic, food, and culinary to biocultural uses and applications. And so, the ethnobotanical knowledge held by this Manubo community in the province of Agusan del Sur, Philippines, is an important resource that should be conserved, documented, and studied. And while the local names of their medicinal plants are useful in ethnopharmacology, reliance in vernacular names for species identification can cause ambiguity and incorrect identification, resulting to research invalidation. And during our expedition and fieldwork, we usually collect medicinal plants lacking reproductive parts without fruits and flowers, so it is hard for us to use morphological identification itself. And so we did molecular confirmation. And here, for my herbarium and laboratory work, I did this during my doctoral degree at the University of Santo Tomas and during our fellowship grant with my advisor, Dr. Alejandro, at the University of Bayreuth, being a junior researcher sponsored by the Alexander von Humboldt Foundation. And here, I am so proud to share our research output published in BMC Springer Nature of the Journal of Ethnobiology and Ethnomedicine, entitled Quantitative Ethnopharmacological Documentation and Molecular Confirmation of Medicinal Plants Used by the Manobo Tribe of Agusan del Sur, Philippines. This study is actually the integrative molecular approach confirming 24 species with confusing species identity using multiple universal markers like ITS, MATK, PSBA, TRNH, RBCL, and TRNLF. And this study recorded the first quantitative ethnopharmacological documentation coupled with molecular information of medicinal plants in Mindanao, Philippines. For integrative molecular taxonomy or confirmation, a combination of a priori data from ethnoclassification and a posteriori data from both morphological and molecular data were used in this study. Ethnoclassification is based on vernacular names and traditional knowledge, and determine the scientific names on the reference of the local names using the Dictionary of Philippines Plant Names by Madulid in 2001. For morphological identification, comparative morphology using available literature and worldwide databases, and with the assistance of an expert and a botanist from the National Museum of the Philippines in the name of Mr. Danilo Tandang. For molecular identification, this study utilizes multiple universal markers, both the nuclear and chloroplast markers for sequencing and blast matching. And so for blast similarity-based identification involving using the simple method, taking the top 5 hits of the max score and optimized approach using the formula. And within a 5-point deviation down to the max score, the highest max score and the highest percent identity were also determined. And all the species identity, generic, and familial affinity were further confirmed from the recorded occurrence and distribution of putative species in the study area based on the updated COS digital flora of the Philippines. And here, I am glad to share another published article confirming 2 species of piper using integrative molecular approach. It can be concluded that species identities can be confirmed using molecular data when morphological data is limited and inconclusive, while constituent and cytotoxic evaluation can be useful in determining variations between plant species used in ethnomedicine. And here in these highlights of the study, molecular confirmation of an indigenous Piper decumanum and an introduced Piper aduncum. This also reinforced the ethnoclassification of Piper species using integrative molecular approach, and the application of constituent and cytotoxicity evaluation for species comparative analysis. And so here are the challenges and opportunities during the pandemic. During the onset of pandemic, I hereby presented the current status of ethnobotany in the Philippines and possible post-COVID-19 changes and opportunities. So you can check this out in the COS Digital Flora of the Philippines Facebook page with the link below, and to be updated with the current status of Philippine ethnobotany. And I am also very glad to share this opportunity granted on me to contribute to the largest book project of Springer Nature in Switzerland, to contribute 20 book chapters on the ethnobotany of the mountain regions of Southeast Asia. Herein, I was able to feature 20 iconic, interesting, and endemic Philippine medicinal plant species, and some of these medicinal plants were confirmed using molecular sequences. So you can check this out in my research gate and request me a full text so I can share it with you. I am also very glad to share these accolades when I won the Best Poster and People's Choice Award in Poster Competition for professional category during the Regional Health Research System of the Northern Mindanao Consortium of Health Research and Development last year. And also, I am very happy to share my recent award in Best Oral Research Presentation during the National Health Research System in Marriott Hotel, Clark Freeport, Pampanga last July this year. I was the only finalist that time representing Region 10 or Northern Mindanao, since I also won the People's Choice Award in oral competition and the first place winner during the Regional Health Research System of the Northern Mindanao Consortium for Health Research and Development. And also, I got opportunities in various media interviews and news features in disseminating our research outputs to local and international news platforms, radio and television interviews. And aside from that, I am also happy to share the news feature of my research study this year in a local press, Mindanews, which is a Mindanao-wide press based from the city of Davao. And aside from the local news feature, I am very glad to share this international environmental news feature of 𝗠𝗶𝗰𝗀, which is a US-based nonprofit conservation and environmental science news platform featuring my published study. And I am also happy to share that I also got interview in Net 25 under episode of ASPN or Ating Sagipin, Planeta Natin during our state universities and colleges fair this year at Mall of Asia Arena Music Hall, together with the CHED Commissioner Prospero Popoy de Vera III, while flexing our SEPA materials from SEBREM, and also the local radio stations from DXGT Radio Abante and DXMU 88.9 FM. With this, we are so privileged to be able to disseminate our research outputs in promoting public awareness of threatened medicinal plants in different mountain ecosystems of Mindanao. And as part of the 6 P's of the project, here are some more opportunities in store as we capacitate the people by educating the locals or indigenous peoples and training the undergraduate and graduate students. And also, we went to several places for fieldwork activities, samplings, and expeditions, including partnerships with the local government units, indigenous communities, and collaboration with the Philippine Genome Center Mindanao, thanks PGC Min, with our NRCP-funded project on the molecular taxonomy of medicinal plants in Bukidnon, Mindanao. And here's more. To continue our future directions for basic and applied health research, drug discovery and development, we have the Tuklas Lunas Development Center under the Philippine Council for Health Research and Development. We also have the DOST Virology Institute of the Philippines, and we also have the National Research Council of the Philippines, the Department of Health, Philippine Institute of Traditional and Alternative Healthcare, and the Philippine Genome Center Mindanao for their funding support, partnerships, and collaborations for achieving our national development. Maraming salamat po, especially to all taxpayers in the country. And of course, special thanks to our first collaboration with PGC Mindanao on our project funded by the National Research Council of the Philippines on the molecular taxonomy of medicinal plants in Bukidnon, Philippines. Thank you very much, Chancellor Lair, Director Joel, and PGC staff and personnel for this opportunity. Maraming salamat po. And here's more, like more funded projects, more collaborations, more field expeditions, more genomic studies, more omics research, and more paper publications. Maraming salamat po, PGC Mindanao, for this opportunity. And of course, we are sincerely grateful to the following institutions and individuals for their help, assistance, and support to make our omics research possible. productive, and successful. Not to mention all the taxpayers again in the country. Maraming salamat po sa inyong lahat. And before I end this, I'd like to end with a famous philosophical thought of a Swiss physician, an alchemist, a late theologian, and a philosopher who pioneered the medical revolution during the German Renaissance in the name of Paracelsus, telling every one of us that the healing comes from nature and not from physician. Therefore, the physician must start from nature with an open mind. Maraming salamat po sa pakikinig. Mabuhay po tayong mga Pilipino. Salamat po.
Thank you very
Thank you
much,
po.
Mark. I think we have a video to be played. Jans, please. Thank you, Doc Mark, for sharing your extensive research on ethnopharmacology. I hope we can do the same in other plants. Go ahead, Jans.
Okay na yan, Ma'am Lay.
Okay. Sige. Okay, so let us now have the open forum. I'm sure our audience are excited in asking questions. May I ask our speakers to turn on their cameras, please? Okay, so let me read the first question for Doc Weng. Wait lang. I think you already answered Ma'am Lynn's question, but maybe you want to elaborate more, or you summarize your answer to Doc Lynn. Her question was, she was curious about the testing on human cells from infected individuals and non-infected individuals. Would you expect uniform responses from different human cell sources? Thank you for elaborating on this. Thank you, Ma'am Lynn, for the question. Go ahead, Weng.
Okay. Yeah, thanks, Ma'am Lead, for listening to my talk. So yeah, as I mentioned in my answer there, so we expect to see different responses from infected versus non-infected individuals, or the immune cells from these individuals, in terms of antiviral gene expression, interferon, other components of the interferon signaling. And we might also expect different results or contrasting results from individuals from different age groups and from individuals with or without the comorbidities, as we consider the profiles of these individuals, including their vaccination status. So we will look at different angles, like we would deal with different data and categorize them. We have Ireland has the, what they call this, a biobank of samples from COVID-19 infected individuals. So they obtained blood samples from these individuals and share it with different organizations, and they prepare the profiles of these individuals and share it with us, so that yeah, we can also consider their profiles, their age, their vaccination status, comorbidities, in addressing our results.
Yeah, thank you.
And to elaborate, she has a further question.
Yeah, so she
Yeah.
said, would increasing sample size or source of the cells provide more meaningful data, or what additional testing or method would be needed to confirm findings?
Yes, definitely. But as of now, we close our number of participants, as
cases now are very few, especially that Omicron is like shows common symptoms such as flu. So like we couldn't recruit more patients, like as few patients go to hospital as of this time. Yeah, since the dominant variant is the Omicron.
So I hope your itchy throat is not that variant, huh?
Yeah, I think it's just a common cold.
I hope. Thank you, Weng. So there's a comment. I think this is more of a comment for Vice Governor Uy, also from Dr. Lynn Ralios. So this is a congratulations, Vice Governor Uy. This is a testament to the impact of collaboration and partnership in providing effective and efficient solution to pressing problems in the community. But I think the most important factor here for Davao de Oro's success in this initiative are the political will and innovative and informed leadership to make things happen despite the socio-economic hindrances and criticism. So would you like to comment also on that, Governor Uy? Vice Gov po. Sorry. Gi-promote na kita.
Thank you. Thank you pala sa nag-comment. I appreciate it. And as sinabi ko kanina, yun nga, may mga backlashes, pero at the end of the day, as a leader, I was mandated by the people to be there to put long-term plans for our people. So sa tingin ko, itong project na ito bigay ng malaking impact. So I decided, bahala na ang Diyos sa decision ko. But I think no regrets. I made the good decision, and yun, naging asset na ng probinsya itong laboratory na ito.
Yes po. So question with regards to that, Vice Gov. Good day from an anonymous attendee. You mentioned the effect of, wait lang. Saan na ba yung tanong ko dito? Ayun. So this is from one of our viewers from FB, no? So now that the COVID-19 cases is low, would the laboratory be open to cater other molecular biology services, especially for the academe and agricultural sector? I think that's also my question, no, Governor Uy. I know that Davao de Oro also have this neglected tropical diseases, no? So can you answer that? Ano pa po yung mga ibang services that we can offer, no, because you have the laboratory already? Do you have
Yes, ma'am. Ang notion talaga dyan sa partnership din natin is to make it a parang research and development extension of the UP Genome Center. So pwede po kung ano yung pwede nating mabigay na serbisyo through the laboratory. Pwede po pag-usapan natin yan.
Yes po. Sayang kasi, sir, di ba? Ikaw, medical technologies din po kayo. I know we have other neglected diseases that we need to focus on. So I think it's a good opportunity for us, no? Andyan na yung molecular lab, so we can make the most out of it. Thank you po. So let me ask another question for Dr. Tapar. Also from anonymous attendee, may I ask the following? What is the difference between folk taxonomy and ethno-classification? Nakabalik na ba si sir? Ayan, I think nahirapan siya sa kanyang connection. Doc Mark, can you see the question po? Here?
I'm sorry, ma'am.
Sige po.
We got a heavy rain. Ano po yung tanong?
Okay po. What is the difference between folk taxonomy and ethno-classification? Are they fall under local ecological knowledge or LEK? Yun po yung first question.
Okay, thank you very much for that question po. That is just simply synonymous po. They are closely related in terms of the indigenous knowledge and native science. So this boils down to a traditional ecological knowledge or system. So you can actually interchange that one.
Yeah. So the next question po is, due to budget constraints, is it recommended to use ethno-classification or folk taxonomy and morphological or traditional taxonomy kasi daw mahal ang molecular methods? Yeah, I think this is really the problem. Sige, sir.
The case po, I cannot guarantee that folk taxonomy could be reliable because that could lead to research invalidation if and when most of this identification is not validated or cannot be supported by its morphology. At the same time, limited yung funds natin for molecular to support that. So talaga hindi natin siya ma-confirm. Yan talaga yung species na yan.
Correct po.
So
Yeah.
I suggest na wag mong asahan yung folk taxonomy.
So we can also use the sir, no, yung mga RT-PCR lab, yung mga molecular lab ng bawat province, no, for this purpose, I guess, no.
Yes po.
Aside from infectious diseases. So meron pa tayong ibang pwedeng gawin. Yeah, thank you po. Another question for Doc Louela. Your study is very interesting. What do you think is the fit educational background or skills we needed to qualify working in such studies? O, kailangan ma-inspire mo sila, Weng. Go on research.
Oh, yeah. So for me, I took up BS biology. So yeah, for undergraduate degree, so you can take any life science courses, could be BS biology or specific molecular biology, or even chemistry, pharmacy, and like take up graduate studies for or to specialize whether in drug development or virus research, do graduate studies on molecular virology or what else? Medical science, something like that. Yeah. So pursue graduate studies after undergraduate, finish the undergraduate degree. So there are a lot of opportunities out there. Actually, even in my case, I'm not a stellar or like a star student in UP Mindanao, but I still pursue graduate studies, and I was so surprised that I was chosen I was one of the chosen Panasonic scholars. Only 3 at that time, only 3 from the Philippines were selected, and I was one of them. I wasn't even like one of the stellar student, but thanks to the support of my mentors, even my supervisors, Ma'am Lynn, Dr. Murao, our dean, previous dean before, Ray Abad, and Dr. Ray Abad and Dr. Rasco, they were very supportive in my pursuit for graduate studies. So I owe everything to my mentors. So yeah, keep in touch with your mentors because they will like help you reach your goals to becoming a scientist.
Yeah, so we are really proud of you, Weng, no? I met Luella when I was also in Japan. Akala ko hanggang master's lang siya, but she was able to secure another PhD scholarship, no, from Honjo. So kayang-kaya natin yan, no? Like
Yeah.
what Luella said. So I hope the students here are inspired, no? There are maraming possibilities, no? Mag-apply ka lang talaga. Sige, thank you, Weng. Another question for Vice Governor Uy. Sir, you mentioned the effect of prolonged isolation to the mental health of your people. I believe this aspect is important and crucial. With this, did you have any program to address this issue po? Especially that this is not only the prolonged isolation, but the pandemic as a whole that contributes to the mental health issues that we have.
Thank you. Kasali po doon sa present ko, ma'am, yung DDO helpline. Andun po ang number din ng opisina ng social welfare natin dahil may psychologist tayo. We call the program Lifeline Program na pwede sila makakonsult doon. So that was the program that we were able to introduce during the pandemic related to mental health issues.
Okay, thank you po. Another question po for you, Vice Gov, from our Chancellor 𝗪𝗀𝗀𝗪𝗀. So the pandemic was just a preview of the emerging challenges in the 21st century. Innovation and technology will remain vital tools for managing disruptions. So how can the academe and LGU continue to work together towards preparedness for such disruptions? Hello, sir.
Well, sa ginawa namin with the UP through Dr. Morao, continue lang sa partnership, sa collaboration, more consultations to each other. It's a leadership, no? But of course, we've got to approach it like a whole of nation approach pa rin, to prepare us better in the future, especially sa mga cases like pandemics, hindi, yung mga man-made or natural calamities. Yun naman lagi naman, no, ang kailangan natin. Opo.
Yeah, thank you po, sir. Question, my personal question to Dr. Mark, no? Doc Mark, are you still there? I saw that you have your study is more of the ethnobotanical, no? And it's difficult to secure an NCIP clearance. So can you share with us how did you deal with this clearances, no? Mahirap po kasi mag-secure. Yun yung laging nasa mind ng mga tao. But yeah, recently we also asked for a clearance. So, but in your case, medyo mahabang process yan, I think. Can you share that with us, sir?
Well, I appreciate Ma'am Alayla that you really understand the tedious work of this NCIP certification procurement.
Yes, sir.
Abot talaga ng isang taon para ma-secure po yan. But what I did was I got a partnership with the local government unit of the provincial government of Agusan del Sur, and with the help of provincial governor, who is so supportive with this particular research, and it's becoming an agency to agency kasi nga yung director ng NCIP of Caraga was also a relative of our governor, so Director Plaza din. So at least mabilis po kapag kaganon. So it would be better that you have to forge partnership with the local government unit or to the provincial government, because they are willing to support, particularly for this research on the ethnobotanical studies, because this will be aligned on their programs. Kasi may mga programs po sila na nag-exist sa kanilang provincial government units. So i-align ko doon para this will be a give and take na collaboration. So you can also help the provincial government, at the same time the government from the provincial administration can also support you in your academic intent for research and productivity.
Yes, thank you, sir. Yeah, kasi minsan 'di ba parang nagiging hindrance 'yun sa atin na mag-research on IPs, no, na naiiwan natin sila kasi because of that tedious work. But I guess and also NCIP is very helpful, no. We recently communicated with the Region 11 office of NCIP. Willing naman talaga silang mag-assist para lang mawala yung iniisip ng mga tao na mahirap pag may ethnobotanical or ethics issues, no? Pero I guess we really need to deal with that because we need to protect our IPs also. Diba, sir? Yeah. So thank you for your answer. For Ma'am Luella also, again, did you do molecular structure studies for either the drug molecules or the molecules interacting with the drug molecules?
I guess this one is for the HIV study I did, because I didn't explain anything about drug molecules in my study for SARS-CoV-2. So for SARS-CoV-2 I didn't do any structural studies, but for HIV also I didn't. But that part is like more of chemistry. I'm into biology. My field is in biology, but I do some protein, what do you call this? Protein, small molecule interaction studies. So different assays for understanding their interaction, but not mainly on the structure itself. Yeah.
Yeah, okay. So I think you can answer also the next question. Tinanong ko na din to sa'yo kanina. Yun nga, yung do we need to create another vaccine to prevent this immune evasive strain? I think we are experiencing it right now. Yung may booster ka na pero nai-infect ka pa rin. So I think it's another, it's a related question.
Yeah, ideally, yeah, it would be good that if we have a new, like a new vaccine that would target these different mechanisms by SARS-CoV-2. But as of our current vaccines are directed to induce immunity for spike protein. So what do you call this? The variants of concern, the current variants of concern are have mutations in mainly in the spike protein. So like somehow it's like enough, I would say enough at the moment. But if like there is a especially that Omicron is just like we are experiencing just mild symptoms for Omicron, and but we still get infected anyway. But for maybe for highly for variants that would pose risk to our health, for example, like causing severe disease. Yeah, definitely we need a more potent vaccine that targets this different mechanisms of the immune response of the virus.
Okay, thank you. So Governor
The
is
innate immune response.
yeah, thank you, Weng. I think the Vice Governor Uy is already answering some of the questions. Thank you po, Vice Gov, kasi medyo limited na yung ating time. So the last questions are directed to Dr. Mark. I think it's still question on the IPs po, no? So I will just summarize it. So how do you deal with some indigenous people who do not clearly share the necessary infos, and what are their common reservations po that you encountered?
That is a perfect question. That is a very good question. Some respondents are sometimes unwilling to share their medicinal plant knowledge with others due to their previous experience being taken advantage by business-related parties of drug and pharmaceutical companies. And it was also observed that most respondents are becoming educated and with the help of the government education programs for IT. So there's no problem with that, which made them more resistant to allowing themselves to be the subject of the study by the visitors or outsiders. But in spite of that, it is very important to gain trust and confidence and respect the tribal communities, the indigenous peoples, the cultural communities, by embracing the rich cultural tradition through ritual observation and tribal immersions within their community. Huwag po nating unahan ng pagtatanong. Kailangan po nating tanungin kung anong kultura na kailangan nating i-observe before we will going to penetrate with their cultural territory. Although they maintain secrecy about their medicinal plant use and knowledge, it is also beneficial, as far as I'm concerned, to practice keeping their knowledge from overexploitation for protection, and of course to keep them from being over abused by other people based on their plant resources and knowledge. So kailangan din natin i-secure din yan.
Correct. So I think this meron kasing memo ang FREB, no. We have a recent case, Dr. Mark, if you are familiar, that there's a big publication involving IPs na hindi approved ng ethics, no. If you are familiar with that, they collected saliva from the indigenous people. So the purpose is not to stop us from doing research, but involving IPs, but we should be really protecting our indigenous people by doing this, by following the protocol of NCIP. Okay, so I think we don't have enough time. It's already 11 o'clock. So thank you very much po to our speakers for your talk and for engaging us with this open forum. Let me turn over now the spotlight to our host. Thank you very much po.
Thank you, Ma'am Alayla, and to our speakers for that very insightful discussion. Now, to award the certificate of appreciation to our speakers, may I ask Dr. Joel to join us? Please turn on your camera po, sir. May I also ask Dr. Alayla and the rest of our speakers to please turn on your cameras.
Yes.
The certificate reads, Certificate of Appreciation is presented to 𝗀𝗁𝗀𝗀 PhD, for being the resource speaker on elucidating antiviral immune evasion mechanisms of SARS-CoV-2 using omics approaches, lessons for the future during the Mindanao-wide omics webinar held on October 28, 2022, signed Joel Hassan G. Tolentino, Program Director of PGC Mindanao, and 𝗢𝗍𝗄𝗪, Executive Director of PGC. We would also like to give you this Mindanaoan gong from PGC Mindanao.
Thank you so much.
Para magkuha niya na.
Thank you.
The same certificate is given to Vice Governor 𝗀𝗻𝗪 for being the resource speaker on innovation, creativity, and change amidst COVID-19 notes and insights from Davao de Oro during the Mindanao-wide Omics webinar held on October 28, 2022, signed Joel Hassan G. Tolentino, Program Director of PGC Mindanao, and 𝗍𝗪𝗄𝗪, Executive Director of PGC.
Thank you. Thank you po.
Thank you, Vice Governor. And last but not the least, Mark Floyd G. 𝗀𝗏𝗏 PhD, for being the resource speaker on integrative molecular taxonomy of medicinal plants used by indigenous peoples and cultural communities in Mindanao, Philippines, during the Mindanao-wide Omics webinar held on October 28, 2022, signed Joel Hassan G. Tolentino, Program Director of PGC Mindanao, and 𝗍𝗪𝗄𝗪, Executive Director of PGC. Thank you, Doc Mark. So all of the speakers will be given this Mindanaoan gong as their token for this webinar. We'd like also to mention the presence of Chancellor Murao, the former PGC Mindanao Director. Thank you po, Ma'am Lar, for gracing our event. And also the certificate of appreciation to Dr. Alayla E. Decadis, PhD, for being the moderator during the Mindanao-wide Omics webinar with the theme Omics in Mindanao, Challenges and Opportunities During the Pandemic, held on October 28, 2022. Signed Joel Hassan G. Tolentino, Program Director of PGC Mindanao, and 𝗍𝗪𝗄𝗪𝗀, Executive Director of PGC.
Thank you.
Thank you, ma'am.
PGC.
So I would like to ask all of these speakers and the research and Development Division team, as well as our moderator, to open their cameras for a photo opportunity. We'd also like to ask Chancellor Murao to turn on her camera. Okay. 3, 2, 1, smile. Again, 3, 2, 1, smile. Thank you very much to all our speakers. So before we go to our closing remarks, we will have another set of questions for our games. So again, the first one to type the correct answer on the chat box will be the winner. So for this question, the prize will be 5 limited edition PGC Mindanao notebooks. So for this question, this comes from Vice Governor 𝗢𝗰𝗀𝗀 presentation. So in which town or municipality in Davao de Oro is the Davao de Oro Biomolecular Laboratory situated? Okay, the answer is Monte Vista, Davao de Oro. So the winner for this is Mr. Romel Steve Mazo. Congratulations, sir. One of our staff will get in touch with you for your prizes. For the next question, how many years now is PGC Mindanao since establishment in 2019? For the correct answer is three years old. We are now 3 years old. So the winner for this is Mr. Paul Placis. Sir Paul, you won the grand prize. For this question, our prize is a discounted price for our training. So one of our staff again will contact you for your prize. Thank you so much for participating in our games. So for future event and to connect with us, visit our website and social media pages. Now to formally close this very special event, let us all welcome again our Program Director in PGC Mindanao, Dr. Joel Hassan Tolentino.
Hello. Maayong adlaw usab sa inyong tanan. Good day once again, everyone. As you already know, today is the 3rd anniversary of PGC Mindanao in reference to its launching to the public last October 28, 2019. As a continuation of our anniversary celebration, for the remaining 2 months of this year, PGC Mindanao will still be very busy as the center will conduct the Bioinformatics Lecture Series, which is specifically hosted by our Bioinformatics Division. PGC Mindanao will also have its caravan in the Caraga region, or Region 13. We will be at the Philippine Science High School Caraga campus in Butuan City on November 25. We invite our partners and future partners there in Region 13 to join us in this one-day event. Furthermore, PGC Mindanao will continue to provide its training and dry and wet lab services and engage in collaborative research. The COVID-19 biosurveillance efforts of PGC Mindanao will also continue. The center will also spearhead in the finalization of the Manual of Operations of the Mindanao Genomics Consortium. We hope that you continue to take part in these activities. We also look forward to the visit to PGC Mindanao of Dr. 𝗀𝗀𝗀𝗀 through the DOST Balik Scientist Program, hopefully this December or maybe early next year. At this point, I would like to express my gratitude to the following. Number one, the participants. Thank you for your attendance, and we hope you learned from our speakers insights that you may apply to your respective omics studies and activities. Or if you are not yet into omics, we hope that you will be one of us. To our moderators, Vice Chancellor and Technical Expert Novero and Technical Expert 𝗢𝗍𝗍, for leading a very engaging discussion during the Q&A. To our distinguished speakers, Drs. Naredo, Dr. Ramos, Humawan from Day One, and today, Drs. Ongo and Dapar and Vice Governor Uy. Thank you for sharing your expertise and experiences. Indeed, you are an inspiration. Daghang salamat sa inyong paggahin o panahon sa PGC Mindanao, sirs and ma'ams. To Chancellor 𝗪𝗀𝗀𝗀𝗪𝗀, VP 𝗀𝗀𝗀𝗀, and Executive Director Saloma for your presence, messages, and undying support to PGC Mindanao. To the MGC members, Mindanao Genomics Consortium members, and future members for the partnership and collaboration. To the PGC Mindanao personnel, both admin, our hosts Jans and Marian, and lab personnel, for your hard work and sacrifices, particularly in the staging of this event and the regular operations of PGC Mindanao. And also to our technical experts, PGC Mindanao technical experts, for sharing your expertise in the achievement of the goals of PGC Mindanao. Thank you all. Daghang salamat, and may we all have a great day ahead. Padayon, Mindanao.
Thank you, Sir Joel. Now to our participants, you can get your certificate of attendance by filling out the evaluation form sent in the chat box. We will close the form after 20 minutes. So once again, thank you very much, Dr. Joel. Thank you so much to our speakers from yesterday and today, to our moderators as well, and to our participants since yesterday. Thank you for tuning with us in our two-day webinar series. Happy anniversary to PGC Mindanao, and this is the Mindanao-wide Omics Webinar 2022. Stay safe and have a great day.